This is the Hardy-Weinberg practice problems video key. So make sure you guys have completed that handout before you look at this video. Number 1, we have a population in equilibrium. And this problem gives you the allele frequency of capital A. So this means that I've given you the frequency of P. So P equals 0.9. Remember one of our equations, P plus Q equals 1. So we can easily find Q. So 1 minus 0.9 gives us 0.1. So now we have our allele frequencies, and we can use these to find the frequencies of the genotypes. Remember, genotypes are combinations of two alleles, so big A, little a, or little a, little a. Remember, to calculate genotypic frequencies, we use the Hardy-Weinberg equation, p squared plus 2pq plus q squared equals 1. Okay, p squared is the genotypic frequency of big A, or homozygous dominant. So to find that, we just take p and square it. So p squared equals 0.9 squared. So to find q squared, we do the same thing with our q value. So q squared is going to equal 0.1 squared, so 0.01. And then 2pq, we just plug in both of those terms. So 2 times 0.9 times 0.1, and that's going to equal 0.18. Number 2 looks at a population of flowers with different colored petals. So this one is asking about if the population is actually in equilibrium or not. So there are 3 steps to this process. So first, we need to find our P and Q values from the given population. Next, we're going to calculate our expected values, and we can do that using the Hardy-Weinberg equation. Last, we're going to calculate the actual observed values from the population numbers, and from there we compare our observed and expected and see if they're the same. On this slide, I pulled out our relevant information. So we have 170 homozygous brown individuals, and if it helps you guys to visualize this, we can look at the notation CB. So that's the genotype homozygous brown. For homozygous purple, we can say they are CP for purple, and our heterozygotes are going to be CB, CP. So one allele brown, one allele purple. Alright, so before we get started, let's figure out the total number of alleles in our population. To do that, we're going to add up all of these individuals. So we have 531 individuals in our population. Each individual has 2 alleles, so we're going to multiply that by 2, and we're going to get 1,062 alleles. So I'm going to write that over here. Now we need to figure out the allele frequencies for this population. So we're going to start by looking at our homozygous brown individuals. There are 170 of them. Each individual has 2 alleles, so we're going to multiply that by 2. Then we're going to look at our heterozygotes, and there are 21 of them, and each have 1 copy of the brown allele. This gives us 361 copies of the brown allele in our population. To make this into a frequency, we have to divide 361 by our total number of alleles. So we have a frequency of 0.34 for the brown allele. Now we're going to do the same thing for the purple allele. So there are 340 individuals who are homozygous purple. Multiply that by 2. Add up our heterozygotes, because they each have 1 copy of that. And now we have a total of 701 purple alleles in our population. We're going to divide that by the total number of alleles. It's going to give us a frequency of 0.66. Step 2 is to calculate our expected values. And to do that, we're going to use our Hardy-Weinberg equation. So p squared plus 2pq plus q squared equals 1. We're going to take these calculated p and q values and we're going to plug them in. So we don't actually know which one is dominant and which one is recessive. So I'm just going to play the numbers game and say that the bigger one is p and the smaller one is q. Okay, so p squared, we're going to use 0.66 and square that. Okay, so that number is going to equal 0.4356. To get our q squared, we're going to take 0.34 and square that. This gives us 0.1156. The heterozygotes, 2pq. So multiply 2 times 0.66 times 0.34. This is going to give us 0.4488. Third step to this problem is to calculate observed values for our genotypic frequencies. So to do this, we're going to go back to the number of individuals in the entire population. So we know that that was 531. And we're going to calculate using the actual numbers we observed. So for homozygous brown, we have 170 individuals. We divide that by 531. And this is going to give us a frequency of 0.32. And when we do the homozygous purple, we're going to see 340 of those divided by our total population, frequency of 0.64. And then the heterozygote, we have 21 individuals divided by 531 in our population, gives us a frequency of 0.04. So now we need to compare these to our expected values that we calculated using the Hardy-Weinberg equation, and we can see that none of these values line up in any way, shape, or form. So because our expected do not equal our observed, this population is not in Hardy-Weinberg equilibrium. So number 3 is going to look at sickle cell anemia. So you guys should remember looking at genetics problems from this disease when you were in 111. So we are given a percentage here. So 9% of the African population is born with little s, little s. So this problem has given us the frequency of the homozygous recessive individuals. So now we know q squared. And from here we can figure out our other terms, or specifically we want to know the frequency of the heterozygote. Let's start by solving for q. So we're going to take the square root of 0.09. This equals 0.3. From here we can solve for p, because p plus q equals 1. So 1 minus 0.3 is going to give us 0.7. Okay. Now we can solve for 2pq. (2pq) equals 2 times 0.3 times 0.7. So we have a frequency of 0.42. This problem asks for the number as a percentage, so we're going to multiply that by 100. So 42% of our population are heterozygotes. Here's another problem presenting us with percentages. So 70% can taste PTC, and remember, being able to taste is the dominant phenotype. Okay, so there are two genotypes that correspond to that. And let's use T for tasting. So homozygous dominant and the heterozygote can both taste. So remember, this accounts for 70% of our population. The other 30% cannot taste, which means they have the genotype of little t, so they are non-tasters. So if 30% are little t, we now know our Q squared. I'm going to turn this into a decimal. So 0.3 is our frequency. From here we can find Q by taking the square root of 0.3. Yes, you're going to need a calculator for this one. This equals 0.548. Now we can find P. P equals 0 minus 0.548, 1 minus Q. So P equals 0.452. Now we can find P squared. So again, you guys can use a calculator here. So we have 0.2043. And now we can find 2PQ. So 2 times 0.548 times 0.452. So our heterozygote frequency is 0.495. So this last problem is looking at relative fitness. So we want to determine which of our lions is more fit compared to the others. So remember, when we look at fitness, we're not talking about how big you are or how strong you are. So the size of the lion, this is unimportant information in this case. We want to know about reproduction rates. So how many cubs did each lion father? Okay, and more important than just that straight number of cubs, we actually want to know how many survived to adulthood. Okay, so if we look at these numbers, we can see that Tyrone down here at the end has 19 cubs that survived to adulthood. And that is our largest number. So Tyrone is the most fit of these 4 lions.